given that the virus can evolve much frequent and much more and much faster, this does mean that the virus factors or the viral factors could also influence the virulence. In terms of the virulence factors, the virulence can also be much more in different populations per se. There are certain mutations within the virus which is known to modulate the replication of the virus, or it could influence the host susceptibility, or it could mask the immune system, or some of those have no any influences. So those mutations are provided here from the different regions. If you would like to know more about a specific mutation in the ORF1 in the PCP region, you may go and read this article from ours, where you will have more information what this mutation is contributing in terms of HIV replication, or how this mutation in the RDRP could increase the replication of the hepatitis E viruses and so on. Now I'm going to give you some examples from our own studies, what we have done in Vietnam. Vietnam is a country and they have reached at least 100 million population per se, and the median age is 33 years of age. The country has a high prevalence of viral hepatitis B. The reason is the country introduced the hepatitis B vaccine since 2003, which means people who were born before 2003 were not being vaccinated for hepatitis B. So those who have been infected, who had an acute infection, had now moved into chronic cases, which means from 2003 until today it's like 20 years. So anyone 20 years and below or above is, 20 years and below will not have been infected. However, their highest risk goes for those greater than 25, 30, 40 and so on. So the incidence becomes higher in terms of the chronic hepatitis cases, hepatitis B cases, who are moving down into liver cirrhosis or hepatocellular carcinoma. 23 people per 100,000 die of liver cancer. Since 2021, liver cancer became the top most cancer related deaths in Vietnam, followed by lung and breast cancer. So what we have done in the past is we have done one study which we looked into patients with hepatitis B. As I've said, the Vietnamese population have higher exposure to hepatitis B and higher infection rates. Therefore, we aimed to ask a question: do we see viral hepatitis E in patients with Hep B? To do this, we need to see how many of those people in a normal population has also been exposed in terms of the seroprevalence, but equally you need to understand the spectrum of hepatitis E infection in patients with chronic hepatitis or acute hepatitis or liver cirrhosis or hepatocellular carcinoma. Therefore, this study looked into 1,658 participants with 340 controls and cases. Using ELISAs, we screen for the seroprevalences and infection screening from the serum. Directly, we look for the RNA positivity of this virus. And then we characterize the virus, which of those genotypes do they carry. If you look into the results, 31% of the population has been exposed to hepatitis E. 45% of the 45% of the population who are having a hepatitis B is even more exposed than a healthy individual. In terms of IgM prevalence, 4% in the healthy population compared to that of 12% in patients with viral hepatitis B. genotypes or the RNA positivity, interesting thing is what we found was patients had the genotype 3, which is not 1 or 2, but it's 3, which does mean that patients acquire genotype 3 through foodborne zoonosis. This made us to go into the second study. Okay, let me finish the first study. The first study clearly showed that there is a high prevalence of HIV in HPV patients. Hepatitis E infection or a super infection aggravated the clinical outcome in HPV patients. Number 3 is HIV infection independently associates with liver cirrhosis as well as severity. Based on the genotype 3, what we observed in the study, we went one step back to understand if there is a foodborne zoonotic events going on. We looked at a study group or a cohort. Okay. So in this study, what we looked at is, we looked at the cohort 1, cohort 2 and cohort 3, where we looked at the liver tissues. The main message from this study is the liver tissues which is sold, the pig livers or the livers from the meat, had 12% positivity. And we had high seroprevalence in occupationally exposed, such as people like meat vendors, pig farmers, or slaughterhouse workers. And yet again, we see 12 genotype 3 with subtypes 3B, 3E, and 3F, which is in circulation. This means that there exists pork meat as a foodborne transmission of viral hepatitis E in this population. We also did the same study in Germany. We looked at all the supermarkets where they sold the pork meat with liver and all the different stuff. And here we again see 10% of them were infested with hepatitis E virus RNA. As you see from Germany, the number of cases has gone or risen to 50-fold. As of October 2023, we already reached 3,773 cases which are reported to the Robert Koch Institute. The study 4, as we said, pregnant women is one of the high-risk groups. We looked at 183 pregnant women in their 3rd trimester and looked at the seroprevalence, but also for the RNA positivity, both in the serum as well as in the stool samples. We had 8% positivity in terms of the exposure, but there were only 4 of the mothers were tested positive for HIV RNA, but they did not have any severe outcomes or any poor clinical outcomes in terms of stillbirth or in terms of low birth weight babies. There is no any associations found in terms of an infection with that of the clinical outcome. Currently, we are doing studies from the north, south, and central Vietnam, looking at the wild boar population. And what we can see from our preliminary data is 15% in rectal swabs, 7% in fecal samples, 4% from livers. And the distribution of the genotypes or the subgenotypes differ in different regions, whereas despite all this, it's only genotype 3 which is predominant in the whole of Vietnam, wherever it might be, it could be a north, south, or central Vietnam. But for Indonesia, it's genotype 4. This is what I assume. We need to confirm from other studies. Maybe 1 or 2 of your students can do this during our internship and so. And if you compare also the distribution of the genotypes, subgenotypes, 3C is not found in Vietnam, 3F is found in both places. And we have also done a workshop on viral hepatitis E from the 4 different global health centers, where we have developed SOPs and harmonized SOPs for the different molecular testing. And all the different students spend their 2 months of their time in Germany to get this certification, followed by pilot studies which is ongoing in Gabon, Ghana, Vietnam, as well as in Guatemala. So I would ask you to go into the PACEUP website. You have a study map recently launched. You can look and click at the country-wise, where you can look at the publications, what we have done so far, not only within the framework of PACEUP, but in the recent years, since 2013. So you may go into the PACEUP website, look into the study map, where you can go click. It's an interactive map where you can look for publications related to a bacteria or a virus or any given pathogen, country-wise or a continent-wise. To conclude, HIV is a public health problem, a global health burden. It's a foodborne zoonotic pathogen. Direct contacts with animals increases the risk. The various animal species shows that there is an expanded spectrum of different animals. Cross-species barriers are existing. One health concept should be considered for a better understanding in terms of the transmission dynamics, equally for the prevention and control of HIV infections. There are some 18 unanswered questions that you could look into in terms of dynamics, in terms of vulnerability in population, different modes of transmission, animal reservoirs, potential risk of contact, is there sequence homologies, how effective are these preventive measures if chronic hepatitis E exists in animal population. There are plentiful of questions that one can ask and use different hypothetically driven questions and experimental designs to answer these questions. I think we came to the end of the talk. I would like first to thank Karthika for inviting me here, and equally the faculties in JEMBER. And obviously there is a lot of funding which came through the German Ministry of Education Research, and equally from the Ministry of Science and Technology from Vietnam for bilateral funding on these studies. And this is a snapshot of our research group a 1 or 2 years back. I would like to leave the floor here for further questions after the next session, and I thank you for your attention. Thank you.
Yes, thank you very much, Velavan, very informative and inspiring talk. But you are still okay, right? You look like so tired. The standing is okay for you? All good? All good? So yeah, okay. Finally, we come to the next speaker. I would like to invite Prof. Dennis Nuryadi to come to the stage. As you know, we would like to first give you a test, Dennis, that you are really... Are you going to say something? Are you going? I think it's better to use the other speakers. Yes, okay. And there is also a pointer over there that you can like move around so that you don't have to stand there. Yes. Operator, can you please give the next presenter the next slide? Yeah. So I have already introduced to all of you. Maybe you would like to say something in Bahasa. And we can do also lecture in Bahasa, but Velavan will not understand at all.
Bisa bahasa Indonesia, cuma nggak terlalu lancar kalau.
Okay, siap.
So this is why I'm doing it in English, but theoretically, the normal things I can use Indonesian.
No, please use English. We will not make Velavan like, oh, it's so boring.
But science is very difficult for me in Indonesian.
I understand.
Tapi kalau cuma untuk biasa-biasa ngobrol bisa.
Yes, of course. So later on, if you wanted to discuss with him later on in Bahasa, so there will be no problem. Also, he's always said, no, I'm not so good in Bahasa, but actually, you are still perfect. But
I try to speak it because, you know, I was born here, so you should keep that.
Yes, of course, you should keep that, yes. Okay, so now, Dennis, I don't want to make another discussion more, so I will just give over the stage to you. So, students and lecturers and researchers, Professor Dennis Nuryadi.
All right, so so Kanika, thank you very much for the introduction. So I think I will just start just for the sake of time. So I will do my own timer just to make sure that I would not take so much of your time. So is it, I cannot say my slides, but anyways. Yeah, okay, cool. So, so the topic today is antimicrobial resistance. So that is what I'm interested in. This is so basically moving from the one pandemic, moving to the next pandemic. So the next pandemic with antimicrobial resistance is one which is currently, basically of a global concern. So it will be one of those pandemics, I think, which will be coming and is already there probably, and will be ongoing in the next few years as well. So what I'm going to talk about today, so just as an outline, so I would like to talk to you about why antimicrobial resistance is a problem, and why, especially in South and Southeast Asia, and also collaboration is important to combat the antimicrobial resistance on a global scale, on a global level. So which strategies can be used to combat AMR, and what has worked and what has not worked, and maybe we can just see what we can discuss later on. So practically this is going to be my take on my views on AMR and what is working and what maybe is missing. So we can discuss it later. So as you know, AMR is a problem, so it causes a lot of deaths. So according to the study from 2019, which was published in Lancet, so it caused over 1 million deaths in 2019, so related or attributed to antimicrobial resistance. And it is projected, if it's going on like this, if it's unstopped, so it is going to go probably up to 10 million per year by 2050, which is actually quite concerning. So if we look at the top pathogens, so a lot of the pathogens are those, if you're studying medicine, so you will encounter these pathogens later on in your daily clinical routine. So E. coli, for example, Staphylococcus aureus, Klebsiella, those are actually one of the major ones, and those are nowadays quite difficult to treat. So why is it difficult to treat? Because we do have antibiotics, right? So we have a lot of antibiotics. If you look at the timeline, so the first antibiotics was discovered in the 1920s. And as you can see, until now, there are more and more substances which has been discovered. But if you have a look at the latest, in the last 20, 30 years, actually we have not had so much progress. So a lot of the progress was done maybe before the 1990s, but then starting from the 1990s, there's nothing new. So most of the newest antibiotics is actually just a little bit modified from old substances which we know already. And the latest one which was approved actually, which is kind of newish, is the sideromycin. So I will go into that later on. The problem is, so if you look at when the antibiotics are being used in the clinical sense, so these are the timelines. On the top line you see when the antibiotic was being deployed, so used in the clinics. And in the bottom line, so in the bottom timeline, you see then where the first resistances were detected. So actually it does not take so long. So you use it, after a few years or after a few months even, you can find actually the first resistances. And the funny thing is, well, it's probably not funny, it's actually quite scary. If you look at it, it's becoming faster and faster. So the newer ones, you use it, and then a few months later or a year later, you will find the resistances. So this is actually a problem. And also, the matter is also complicated because, of course, the resistant bacteria is going up. So there is an increase of antibiotic resistance in bacterial infections, or bacteria causing infections. But the number of antibiotics generally, which is discovered, which is developed, is going down. So it's going in a different direction. So it's really actually not so great. So if you look at the pathogens which are commonly causing infections, so which are the most critical, so as you can see here, the red ones on the top, so it's a bit small, but I can probably read it from here. For example, Acinetobacter baumannii, Pseudomonas aeruginosa, so especially those with carbapenem resistance, so really strong beta-lactamase. And also Enterobacteriaceae or Enterobacterialis, which is carbapenem resistant, also resistant to third generation cephalosporin, so the ESBLs, as you might know it. Yeah, so if there was wondering, since I will be talking to you guys here in Indonesia, so I would be interested in looking at the numbers, because it's always good to see. So actually, if you look at it, this is some countries which is in the South and Southeast Asian region. So a study which is actually quite recent, from 2023. You can see that India is leading, but the number of population is also much more. But Indonesia is actually not so far behind. So we have a lot of sepsis, or you have a lot of sepsis. a lot of bacterial infections, and also quite a lot of resistances, also associated and also attributed. And if you look at the number of E. coli, for example, so in the last few years, it actually is kind of increasing. So it started with 70%, and now it's up to 75%. And even the Staphylococcus aureus is also, there is a decline, but it's still quite high, so it's about 40%. So it's actually quite alarming. So this is the numbers I actually found for Southeast Asia as well. So if you look at the carbapenem resistant ones, so it's actually like almost touching 25% in gram-negative, so it's actually quite high. So for you as a comparison, it's actually among the highest in Southeast Asia, along with Myanmar, Philippines, and Vietnam. As a comparison, I can tell you the numbers for Germany. We have below 3%. So it's quite far. So, but why is it interesting for us or for me, even if we don't have so much in Germany? As you know, nowadays we can travel within a day to anywhere. So yesterday we traveled a lot. So with the COVID pandemics, travel is one of the factors which is contributing to the spread of bacteria or viruses. So this is also the case with antimicrobial resistance. So when I started my clinician residency, there was not so much cases of multi-drug resistant bacteria. So ESBL maybe was there, but that was it. So when I studied medicine, I did not know anything about carbapenem resistant bacteria because it was not there yet. It was not discovered yet. But since then, as it was like 2009, I think, when they first discovered it, or 8, until now. So we have a lot of cases actually. So there are a few studies. So this is why I'm interested in that, because of course we are connected somehow globally. So it's not a local problem. So it is connected. So for example, this is a study which is done by the Dutch group in Europe. So they were actually looking at people who were traveling to South Asia, so to India, for example, or to Southeast Asia, or to the tropics generally, and looking at how they acquire ESBL, so extended spectrum beta-lactamase producing Enterobacterales. So what they found out actually, that a lot of them, so 34% of the travelers actually acquired ESBL through travel. So they were negative before and they were positive when they come back. So it's actually quite high. So if you look at the incidence, the highest incidence is in South Asia. So it's basically 7 per 100 person days of travel. So it's actually quite high. And if you can see in the picture, so the redder it gets, the highest is the risk. So the risk is highest in India, but also Southeast Asia is also among the, I would say, the hotspots as well. And why is this relevant for us? Because, so when they return, basically what happened is that they can also remain positive for the next months. So about 11% of those who were actually acquiring ESBL during travel, they still keep it after 12 months. So it's a long time. And then we can, in this study, they could also show that there were also some transmission between people who were traveling and people who were not traveling. So actually, that's quite alarming. A second study from Finland, and they also basically do the same thing, a similar design. So, but what they did here is that they actually monitor the people. So they were traveling into Asia, and then they basically take every day a swab of the person to show when did this person get positive for ESBL and how long, and do they change. So as you can see in the graph maybe here, you can see that, so once someone actually becomes positive or becomes colonized, for example here with E. coli or with Klebsiella, for example, you actually get colonized for multiple days, for a long time. And sometimes you lose it, sometimes you don't. And what is interesting is if you look at that, so there are multiple resistance genes which actually gets carried also, so it's not only one particular gene, it could be multiple genes, could be different variants of the genes as well. And sometimes, in some cases, there are also people who are actually transmitting, so there are also an exchange of this bacteria between the people who were actually participating in the study. So because of that, this is why I think it's quite interesting to look at it on a global level. So nowadays, you have heard about the concept of One Health. So the concept of One Health, so you're looking at different drivers and different factors which are facilitating the evolution or the development of antimicrobial resistance. So one case is, for example, of course, the environment, so environmental pollution, human factors, because the antibiotics we're using in the clinics, in the human consumption, and also in the animal. So because we do use a lot of antibiotics for food production, for example, because antibiotics can also help keeping the animals not only healthy, but they grow faster. So it's an economical side effect, I guess. And what we do is there is a lot of factors also in the society. So for example, also the education, and also just knowing what to do. For example, hygiene factors. So those are a lot of things which are actually need to be considered actually when you look at the global aspect of antimicrobial resistance. So a lot of the things which PACEUP is doing, for example, is doing capacity building, communicating, collaborating. So actually quite on the right track, I guess, with that. So to look at some of the strategies which was proposed to combat AMRs, I took this from a Lancet paper from this 2000. It's kind of like disappearing a little bit, but it's fine, right? So what they suggested is actually to improve awareness, for example, to improve understanding, and also to strengthen the knowledge, reduce incidence of infection through sanitation and hygiene, in prevention measures, to optimize the use of antimicrobial medicines in human and animal health, and also to develop some economic case for sustainable investment so that there is some kind of development for new drugs, new diagnostics, for example, which can come in. So these are the factors which they actually suggested, so we can discuss it later if these are working or not after my presentation. So there is always a high correlation between antibiotic use and the emergence of multi-drug resistance. So if you look at this graph, so these are the data from Europe on Streptococcus pneumonia. So looking at the penicillin resistance, so if you look at the, so on the y-axis, so we have the proportion of the resistant bacteria, so non-susceptible Streptococcus pneumonia or pneumococcus, and on the x-axis is actually how much penicillin they're using in outpatient settings. So if you look at the graph, so the more you subscribe, the more you use, the higher is actually the resistance rate for Streptococcus pneumonia. So there is a clear correlation. And a lot of the antibiotic which is used, if you can see here, is actually a lot in the upper middle income countries and lower middle income countries because there are different factors, I guess. In high income countries, you probably cannot get it so easily. For example, in the US or in Europe, but in some other settings, you might be able to get it easier. So there are some kind of discrepancy here, which might be able to be optimized through various measures. One example which is probably quite interesting to see is colistin. I don't know if you know what colistin is, but this is why I have this slide. So for those of you who know it, then it's just a refresher, I guess. It's one of the antibiotics which can be used actually to treat multi-drug resistant bacteria. So actually we use it as a last resort antibiotic. For example, if it's resistant to carbapenems, to meropenem, for example, this can be used. So it belongs to the polymyxin antibiotic groups. So what it does is actually it binds to the lipopolysaccharide of the membrane, as in the membrane of the gram-negative bacteria. And by doing that, it causes membrane instability and causing the bacteria to break down. So this is actually quite useful. The only thing is that it's actually quite toxic. So this medication is quite toxic. So the alarming thing is that actually there is a lot of countries who are using it. Even in Europe, it is used actually. And so there was this study actually quite interesting who discussed actually about the international trading route. So how the colistin is moving from one country, produced in China most of the time, and then there are intermediate countries producing them or manufacturing them, for example, into human consumption, for pharmaceuticals, also for drugs, and also for additive for animal feed as well. So if you look at this, this is a slide which for Vietnam is actually quite nice because it's kind of a shock because we were in Vietnam the last few days, so they have a lot of usage of colistin, so actually where a lot of pig farming is being done, colistin is used quite often because it makes the pigs actually grow faster and they do not get sick. But also I think in Indonesia it is also used in some cases also for chicken farming, so that you can use it for a lot of things. So, but you can see here, so there is some kind of, still some kind of problem, but why am I showing you this? Because this is, if you look at the graph, Vietnam has a high consumption in both pharmaceutical, the blue line or the blue bar, and also as a feed additive for animal. So if we look at our study, which we did on Klebsiella pneumonia, which were carbapenem resistant, so a study which we did in Vietnam, in Hanoi, within its VG care framework, we actually found that 40% of the carbapenem resistant Klebsiella pneumonia, which we isolated from patients, so clinical isolates, so from infections, they were resistant to colistin. And if you look at the numbers which I have here, so most of them actually had exposure towards colistin. So it's actually quite alarming. It's quite huge. So yeah, what can we do about it? So this is what they did in China, because in China they had a lot of fig farming going on, and they were actually the first ones who were describing that there's a widespread of colistin resistance in gram-negative bacteria. So what they did was they found out, and they banned the usage of colistin. So the question is it working? So by reducing it in the animal consumption, is it working actually generally? So this study is actually quite interesting because it actually shows exactly that. So they found out the problem in 15 and 16 basically. So the study was published in 16, and then from that they started the intervention by banning the usage of colistin. And as you can see, the sales go down, the production go down, so it's going quite well. And then you can see actually in the next slide that the resistance rate is also going down. So the darker the color is, the higher is the prevalence of colistin resistance. So the top part is for chicken, the green part, and the red part is for pig farming. So in both sectors, you actually see quite well that it is getting lighter, the color, so it's actually the resistance rate is going down. So it seems to work. It's not probably not going to solve everything, but at least it's a start. So a second example which I would like to share to you is probably this MRSA, Cillin-resistant Staphylococcus aureus. If you look at the graph, you look at the various different colors. So what is actually quite striking is that if you look at Europe, we have actually a very low rate of MRSA compared to everywhere else, even to the US. Isn't it strange? So what did we do? What thing we did? Are we doing something right? Are we doing something wrong? I don't know. Or maybe we don't have enough patients, could also be. But if you look at the data quite closely, this is a study which we did when I was still in Heidelberg, and actually we saw, so just look at the light gray line. So the MRSA rate is actually even there going down. So actually from 10% in the early 2010 to basically almost 5%, and now we are actually below 5%. So it is actually decreasing. So what happened? If you look at the data in the United States and United Kingdom, so in Britain and in the US, so Britain is the yellow one, and the US is the gray one. They're quite stable, and the UK is actually going down. So what happened? So in Europe, what we did is actually the Dutch actually started it. So what they call a search and destroy policy for MRSA. So basically we search for them and we try to eliminate them. So searching by, for example, doing screening. So patients who are coming into the hospital are being screened. So we took nasal swabs from the nose, for example. Of course, there are risk factors which you actually have to consider by swabbing. So once we know that someone is colonized, so we try to decolonize. So using, for example, topical antibiotics, antiseptic washing, for example, and also patient isolation, and molecular typing. A lot of communication was done, why are we isolating patients? So this is what happened in Europe. So, and from then, there is a good decline. So it's in the Netherlands where the first cases were actually promising, and then it spread out more towards Europe. But I don't know a lot of countries who are doing that at the moment other than the European countries, because we look at our neighbors. We always try to learn from our neighbors. And the funny thing is in Europe, actually, the further north you get, the less antibiotic resistant you have. So we're learning a lot from the Scandinavian countries and also from the Netherlands, actually. So they did exactly that in Denmark. They exactly did the same thing, and it also works. So this is just showing that sometimes like a more, I would say, aggressive strategy might work. So for the gram-negative, it's a bit difficult because you cannot decolonize, because they're in the gut, so it's really difficult to decolonize the gut. You shouldn't. You should also. So this is why it's a bit tricky. But at least from MRSA point of view, this is successful. For gram-negative, we're on the way. It's still difficult. It's still a challenge. So the third part, as a potential strategy, is what I would like to say, understanding the resistance mechanism. So I'm very interested in antibiotic resistance, and not only on the epidemiology, but only on how the resistance is actually developing, how it's evolving. So this is just some kind of an overview. So if you look on the left-hand side, so you have the blue side basically. So this is where the antibiotics are kind of interfering with the processes in the cell. So for example, you see the cell wall synthesis, the protein synthesis, nucleic acids. And on the other side are the resistance mechanisms which you can find for various antibiotics. So for example, the bacteria can pump something out. So by increasing the expression of the so-called efflux pump, so the drug can go in, but it pumps out again, so it's not really working in the bacteria. It can also inactivate, for example, by producing enzymes, for example, the beta-lactams. It can also bypass the target by alternative pathways, for example. And it can also modify the target for the drug itself, so it cannot bind and it cannot work. So the bacteria is actually quite clever, and they can combine a lot of things. So why am I sharing with you this slide or these thoughts? Because actually, we just need to think about actually what happens when we give antibiotics. So how does the bacteria evolve? Some bacteria can do something else, can find other pathways, can bypass. But for example, not every. So I would say the antibiotics which is causing mutations in the bacteria is probably those who are actually in danger of driving the resistance in the clinical medicine, because of course due to exposure it will mutate. But if the mutation, for example, or the resistance is caused by the acquisition of a gene, the gene has to come from somewhere, so it doesn't appear out of nowhere. So these are the two things which one need to think about actually for the next few slides. So there are a few resistance can be acquired from external, so from different bacteria, from a resident, for example. But some of them can just be intrinsic, so the bacteria might have already everything they need. They just need to change expression, just to mutate a certain protein, and that could work. Okay, so why is it relevant? So for me, because I come from the clinical side of things, so for me, knowing the resistance mechanism in the clinical sense is actually quite useful. For example, if I know the beta-lactamases, for example, then I can probably kind of predict if the presence of So if the presence of a certain beta-lactamase, for example, is predicting the phenotypic resistance in my bacteria of interest, in the bacteria which is causing the infection. For example, the beta-lactamases. So there are two groups, two bigger groups. You can divide it into the serine type beta-lactamase and the metallobeta-lactamase. So metallobeta-lactamase is the strongest one, so it can degrade also basically almost every beta-lactam, so the meropenem and imipenem, for example, doripenem and ertapenem. And the other ones, the class A, class B, and class D, class A, class C, and class D, sorry. So they are more narrow and they sometimes can be inhibited. So for example, if I know that in a patient I have a bacteria which is producing, for example, the New Delhi metallobeta-lactamase, so NDN, which belongs to this side, so the class B, that means I can actually also say that actually the bacteria would not be susceptible towards newer antibiotics. For example, ceftazidime avibactam or ceftolozane tazobactam. So because avibactam and tazobactam are not able to inhibit the activity of this particular enzyme. So this is actually quite interesting. Then by doing that, automatically you might be able to reduce also the unnecessary use of antibiotics, because if you know it's not going to work, then why give it? Because by giving it, you're also creating much more damage, because you should not actually forget that actually if you're looking at the bacteria, it's not only one bacteria you are actually treating. The whole body, human body, it is made up of a lot of bacteria. So basically you have a microbiome. So what happens is they can also interact with each other. So microbial communities can interact with each other, for example, by protecting each other. So you have a community of different bacteria. So one of them can produce an enzyme, but they can protect some of the other bacteria which is living in the surroundings, for example, so that the antibiotic concentration can get lowered. And then it could also basically drive some mutation in the other bacteria. So this is the problem why I said, you cannot really see one bacteria as a problem, so you have to see the whole picture actually. And even then, for example, as you know also maybe, so for those who are quite advanced in the clinical medicine, if you have, for example, infections due to prosthetic joints, for example, or anything which is involving plastics or metal, for example, it could be that the bacteria is also producing biofilms. And in the dentistry, for example, they also have biofilms, a lot of them. And these biofilms can also protect the whole community. So bacteria which is inside the biofilm might not be accessible for the antibiotic. So it might also be less efficacious, for example. And a lot of them, so this is maybe getting more advanced into the bacterial genetics, so a lot of them are also having some kind of mechanism to communicate with each other, so through polymer sensing, for example, so sending molecules to kind of like reducing or increasing like cellular stress into different bacteria. So this is actually a lot going on here. So this is why I think it's difficult sometimes to always just to look at one particular picture. So I'll just flip the next slide, it's the same thing. So when we are actually at it, so the question is then, you need to basically just know then, there is also bacterial evolution. This is something which we know from the textbook. We know on the right-hand side, how antibiotic resistance can occur. You have a subpopulation which develop resistance and it can spread. It's a typical selection pressure. But also one thing which you should not ignore is that, sometimes the gene itself is not there, but you might have to acquire it from somewhere else, and it could be, for example, due to transmissions. So this is why this search and destroy policy in Europe is looking at colonization, isolation, so to inhibit or to prevent the spread of bacteria between patients, between healthcare, and also between the hospital environment. So it's actually probably I would say like one success story which is interesting to share with you. So in the beginning I said to you that that bacteria can evolve very quickly. So this is actually one case which we had. So we received this medication which is called cefiderocol. This is a new antibiotic. It belongs to the class of cephalosporin. But it's a new cephalosporin. This is a so-called sideromycin. So what it does is actually, it is a cephalosporin, as you can see from the structure, but it is conjugated with a siderophore. A siderophore is a molecule which is actually binding into iron, so Fe2+, and can go into the ion channel into the cell. So the ions are important for the cell, so it's important for the cellular function. So basically what it does is it's like a Trojan horse. Binds to the ion, it goes into the cells, and then once it's inside the cell, it gets released and it has its activity. So it's actually quite interesting. So this is a new and a very promising drug. So we thought. So we got this, we got our hands on this in the clinical phase trial before it was actually approved by the European Medical Agency. And what we did is that we used it, of course. So we had a clinical case. So we had a patient, 58-year-old. He had a liver cirrhosis for about 10 years. So now he has his second transplantation. So he was transplanted a few years back, and then now he needs another transplantation. So the problem is he developed a bacteremia with a carbapenem-resistant Enterobacter cloacae. So with an NDM, so with a strong beta-lactamase and OXA48, so 2 beta-lactamases, and also with a VRE infection. So initially, we treated this patient with meropenem and colistin. It was not working, so this is why we stopped it.
About 5 minutes.
Yes. And I'm almost finished. And it was discontinued due to the toxic side effects. So what we did is that we then started the new antibiotics. So what happened? So once we started it, so it was okay. So within the first 20 days, everything was fine. The blood culture was negative, but on day 21, he developed fever again. and the blood culture is positive again. But this time, as you can see maybe here, so the staphylococcus is here in this line, it was susceptible, but now it became resistant. So it became resistant actually within 21 days of therapy, which is really unknown because until that time, there is no data on this. So we sequenced it and we actually had a look. So it could mutate, so the siderophore receptor is mutated so that the antibiotic is not really entering this system anymore. So because I thought, okay, that's strange, can we reproduce that in the lab? So we perform a in vitro validation by serial passaging, so increasing or doubling the amount of antibiotics in the liquid culture for every day, so daily serial passaging. So yeah, we did that, and increasing up to 128 milligrams per liter, which is quite high. So what we do see is actually then that here. So this is actually quite simple to see probably, I hope. So the bigger the zone of inhibition, the more susceptible is the bacteria. But here, after a few days, so around day 13, you already see a little bit of small microcolonies growing, and on day 6 you do see it quite well. So actually, on day whatever, 1, 2, 3, 4, 5, so the second week. So we can also actually reproduce this in the lab quite fast. So what happened? So we look at it, and then we actually saw that only those who are producing the so-called NDM type beta-lactamase, so the new Delhi beta-lactamase is actually developing the resistance. The others are not. So if you look at, for example, oxafodate producing ones or the KPC producing ones, actually nothing happens. So they die after a few passages. So what happened? What did we do? So we then performed an inhibition. So we inhibited the activity of the enzyme. And then we can see that by inhibiting the enzyme, actually here, so the DPA is the inhibitor which cannot be used for a human because it's toxic. But by inhibiting that, we can see that actually that the emergence of resistant bacteria can be inhibited. So practically, we know now that, for example, so a certain gene can actually induce a different other type of mutation and causing a collateral resistance, basically. So this is why I said before it's quite alarming and how a bacteria can adapt, because this was unexpected. And now a lot of people are actually reporting this also. So we're confirming our findings. So what's next? So what is quite interesting, maybe looking at phage therapy could be interesting. So this has been done in a few countries. Just want to put it into the room for discussion. And also modulation of the microbiome. For example, you can, do a fecal transfer, microbiota transfer to treat certain things. So these are the aspects which are actually done nowadays in our setting. So now I'm at the end. So to sum up, so AMR, as you can see, is a global problem, and we also need a global effort to somehow find a solution for this. There are a lot of possibilities to combat AMR. We just need to find out which one is working for us. Like I said, is it a search and destroy, or is it the usage of antibiotic? So I think there is no one solution fits all. So it has to be adapted and tailored into the country or into the region you're working with, the setting. And I think in a lot of the cases, prevention actually is the key. So you have to work on the prevention before it gets spread, because if it's already spreading, it's a bit more difficult to contain. So with this, I thank you all for the attention. Terima kasih, und we can discuss.
Okay, thank you very much, Dennis, for very informative and updated information about the multi-drug resistance. And then the next session is the discussion session. For this, I would like to invite both of the speakers to come to the stage so that everybody can see directly. And you can have some movement along. Okay. So please.
Is there any seating arrangements?
It's up to you.
I don't know.
Maybe left and right is, and then I will be in the middle.
Okay. Shall I go on that side?
Okay. Yeah. So now we come to the discussion session. So I would like to invite for the first session. I hope it will be because sometimes one person can give more than one question. So I will limit to 3 questions. I don't know, from maybe 1 or 2 persons. Please, don't forget to mention your name, from which faculty, and so on, as usual. Yeah? From the other side, no? So yeah, 1. And then from this left, okay. 3 all from the right side. Okay, maybe first one, is that Dave? Yes, okay. And the one one with the white hijab, yeah, the next question. So anybody can help me to hand over the microphone? Okay.
Okay, thank you for the time. My name is Dave. Thank you, Professor Falavan and Professor Dennis. It's an honor to have you here. On behalf of my university, I would like to thank you and express my gratitude. And I have two questions. The first one is, it's for Professor Falavan. What's your opinion and fact regarding the great limitation to human? Because we know that usually when it comes to pandemic and infectious disease, it is correlated or in correlation with the zoonotic. We know that Ebola virus is coming from the bus meat, and then Nipah virus is coming from the date palm, and then MERS virus is correlated and from the camel livestock and yada yada. And what I want to ask is there any like data or changing policies that is supporting and that is conclude that limit the bridge between the human and animal is a good thing to do. Remembering and we know that we have a lot of appetite regarding to meat. That's the first question. And the second question will be for Professor Dennis. And I heard you talk about the hotspots in Asia, in Southeast Asia. It is specifically in India and Indonesia regarding the ARR. Are we talking more about only the use of antibiotics or more? Because remembering the One Health aspects, is there any like real example? What are the factors that is correlated with India and Indonesia regarding the ARR? What we need to do as a clinician and a future researcher? Thank you for the time.
Okay, thank you very much, Dave, for your information. Dave will be the one who will come to be the next semester. Okay, yeah, and then the next question from with the white hijab, yeah, okay.
Thank you for. Terima kasih atas kesempatannya. Thank you dan oke. Selamat pagi, good morning to the welcome. I'm Aisyah Nabila Hairunisa for Medical Faculty in University of Jember. First, please me to ask in Bahasa.
Okay, yes.
Thank you.
I will try to translate.
Thank you very much.
But not in, in German maybe or in English. Okay, yeah, please.
Yes. Sebelumnya, kami di semester pertama telah mempelajari tentang berbagai genome editing. Jadi pengeditan dari genom dari manusia itu sendiri, dan itu menjadi salah satu alternatif pengobatan di masa yang akan datang maupun dari segi diagnosisnya. Kemudian di sini juga tadi telah disampaikan bahwa terdapat beberapa cara dalam mengatasi hepatitis E, kemudian yang resistensi antibiotiknya, dan apakah dari segi pengeditan genom dari manusia sendiri terdapat cara alternatif yang dapat diterapkan di masa yang akan datang dari penerapan yang telah disampaikan tadi. Kemudian yang selanjutnya pertanyaannya, di Indonesia di sini kan terdapat isu etik juga. Nah, kami juga sudah mempelajarinya.
Isu apa? Isu apa?
Etik.
Etik, oke.
Iya. Nah, di etik itu bukan hanya dari segi yang internasional, melainkan dari segi agama juga. Karena kami di sini juga menerapkan agama dengan cukup baik ya, dan dengan baik, dan di situ juga ada halal haram. Dan apakah nanti jika diterapkan ke depannya, apakah ada bisa memungkinkan untuk yang halal digunakan, begitu. Jadi dua pertanyaan tadi. Terima kasih.
Sorry, Nisa, can you maybe, lebih spesifik halal haram dalam hal apa? Penerapan vaksin atau antibiotik atau apa?
Dalam genom editingnya tadi.
Oh, masih direlasikan dengan genome editing.
Iya, selain pertanyaan lanjutan dari yang pertama tadi.
Okay. Okay, yeah, thank you very much for the question. Yeah, I think we first have to answer this question because we have already 3 questions, actually. Yeah. So you know the first one, it was more into the policy thing, zoonotic. Yeah. I shouldn't repeat the first question, right?
Yeah, I believe if I understand the question, it's more like the transmission of this emerging and re-emerging infectious diseases obviously shows very clearly that there is animal compartment is clear that the transmission dynamics is taking place, starting from SARS-CoV in 2003 and so, and including the MERS-CoV in 2012, where the camels or the intermediate host. Equally, the Nipah virus is also the bats, and the COVID-19, it also all clearly indicates increased vigilance and surveillance in terms of understanding the transmission dynamics from an animal perspective is equally important, and this has been considered by the policymakers, bringing in like interdisciplinary studies for understanding the transmission dynamics of this emerging and re-emerging infectious diseases. This is in limelight in the last few years, and it's getting more and more increased attention towards One Health. Then in terms of the global health perspective, One Health is equally an important compartment, looking equally also on the environmental perspective, because hepatitis E would fulfill all the three compartments, because you have the animal compartment for the fecal oral route. Fecal oral route, it's basically the environment will also come into play because of the contamination in the sewage and all these different water systems. Humans is there. To put it short, yes, the policymakers are making sure that the One Health is one main, the content that has to be considered and should be addressed.
Yeah, I mean, continuing the question from Dave, I mean, there should be, I mean, it is almost impossible like to limit interaction between human and animal. I mean, what do you think about it? It's almost impossible. So, I mean, how the policymaker, is that really like, if there will be someday like a real like regulation, so do you think would that be really like give a real impact on the zoonotic disease?
Yeah, if I get it right,
Because
so which means like you cannot exclude or like you cannot remove animal companion from your population. So, but where are these things comes from? These comes from foodborne zoonotic things, like these are exotic foods that you eat. For instance, the Ebola transmission in 2015, 2016, this is from one region in Liberia, close to Sierra Leone. People convened for a family function, and they ate bush meat, and this is the focal source of the transmission. Like this is also the availability of less and less protein makes people to go into resources of other food resources, such as taking this different bush meat, but also the meat from wild animals, which increases the risk of bringing from into anthropogenic perspective, from animals to humans, from wildlife into community. And equally, this is also important on an AMR perspective as well.
Okay, before we go to Dennis, maybe finalize with the genome editing, or the one question from the girl, Bian. So she asked about how is it related with the genome editing? Is that applicable to be now to treat like the hepatitis A, maybe for you specifically? This genome editing thing is there coming like the last 10 years, I think.
So far, to my understanding, I have not seen any one single genome editing for a community basis or like an overall basis. Dennis may add up later to this. In terms of treatment, so there is only two things. One is prevention and control. So prevention is better be done in terms of a vaccination. Therefore, you can control the spread of the disease. There is no any treatment options for, I can speak only for hepatitis E, I'm speaking about. So while hepatitis A, you don't have any treatment options, although you can use ribavirin as a potential treatment option, which is also used for hepatitis B. You have a vaccine which has the highest efficacy for 96 to 99% over a period of follow-up of 24 months. So once you implement such a vaccine, you don't have a transmission, or you can block the transmission. Therefore, you can aim for an elimination and an eradication, like polio virus, for instance. Polio is the best example for the elimination and the eradication, what we have achieved in the last 10 years' time. In terms of genome editing, I'm not sure this CRISPR-Cas and all this different are being used in terms of diagnostics perspective. Cas12 or Cas13 based for RNA or DNA things. These are being used in diagnostic perspective, also during COVID times it's being used, but in terms of user friendliness, but also in terms of applicability and readiness to use, this I have not seen much in terms of the clinical routine perspective. Over.
It's not yet, maybe. Yeah, maybe. Dennis, I think you didn't have to, I don't have to repeat the question, right? You
So maybe for the first question was the AMR with the One Health. Yes, I think it's a very interesting question because I think there is a lot being done actually. It's not there's not being nothing being done. For example, if you look at the hotspots, like for example, the pharmaceutical production, especially in terms of antibiotics, is concentrated in the whole world in the regions of India and China, for example. And what happens with the waste? Is it really do we really control, do we really check actually if the water, everything is coming out, is it really completely clean? Even if there are residues, and the residues might not be the whole antibiotic, but it could be metabolites of the antibiotics, it might still have an effect. So there are a lot of efforts being done. So I think a lot of the easiest part to look at One Health is the clinical part, the human, and maybe a bit of animal. But I think the environment is quite important, and that is actually one which is quite neglected a lot. There's a European consortium looking at how much antibiotic and how much bacterial genome is also going into the seawater, for example. So from the hospitals, from the fresh water, from the seawater. And something like this in Asia has also been done in Vietnam a little bit, but I think it's also a challenge because it's a huge program. So for Indonesia, I don't know. I don't think there is such a program. I don't know. I think there are some studies, I think in the University of Indonesia, Universitas Indonesia, they're doing something with colistin in chicken and in chicken farming. But I think in terms of environment, there's nothing being done. And that is actually also one of the agenda I have here, because maybe this is something we can do, because if you have here a focus with agriculture, because that's also kind of soil and everything. It interacts also, of course, with the whole human and animal population. So maybe we can discuss that. So on the second part, I think I can come from the ethical part, from the clinical medicine part. It's difficult and it's challenging. Because I know a lot of them, of course, like you said, religion and cultural point of view is a problem, and I think we need, or we have to respect that, of course. And even in Europe, until now, it's mostly experimental. For example, for HIV, there are some I would say some strategies to kind of do genome editing for receptors of everything for the HIV, but it's not being used clinically yet. And I would say it will not go into the mass use in the next few years, because there is an ethical challenge. The question is when are we allowed to manipulate human beings? Because if at the moment we are having problems with manipulating pathogens, and even manipulated pathogens. So, for example, like what I show you closely, like the microbiome editing. So this is what we call microbiome editing. So we edit just as bacteria to modulate it, and that is a problem. So it's not going to human cells yet. And then if you look at also phage therapy. The usage is very difficult, because then you don't know what happens, and genome editing is really it's a black box. I think until we have further data, I think it's quite difficult. There are a few cases where you can use it, actually, exceptions. So if you basically have no other choice, and then the patient says yes. So if they give their consent and they said, okay, there is no other way to whatever, he will die anyway or whatever, and this is the last option. If he agrees, you can do that. But otherwise, I don't think it will become like the next best thing, at least not in the next 5 years, I guess. I think a lot of the ethical and the culture, that has to be kind of clarified first. And I don't know if you have it here, but sometimes we have some patients with a certain religion, for example, where they do not allow a blood transfusion. So that also we have to accept. So I think as a doctor, you cannot choose. Of course, we always think, okay, we would like to do the best possible care for the patient, but if the patient does not consent, I think we cannot do that without the consent, because that will be unethical. Yeah.
Maybe just point. But so this is like, you know, like in 2000 when cloning came into existence, like Dolly was there, and then you had the next Dolly, and it was short-lived. This mRNA vaccines, this is another example. Some of them do not want to take mRNA vaccines because they where these vaccines decay. Yeah, so there is ethical things that one has to be considered. Also, the genome editing is one big thing which is in the exploratory phase. We do not know how best it can be translated into, into practice or in terms of diagnostics or in therapeutics or whatsoever.
Yes,
It's
please.
actually quite interesting because I think if you look at in the past how a lot of alternative and strategies are happening or being approved for clinical use, it starts with always small cases, 1 cases, 2 cases. It becomes more and more and more, and the more people use it, the more you see that maybe it's not so dangerous. Then the more it can establish itself. I think this is a lot of the cases like that because the regulatory bodies in a lot of countries actually they say, okay, if you want to use this, give me some evidence on how to use it. So this is also important that someone actually uses it. But I think it's not so easy to push, and I would not say probably it is not going to happen in the next 10 years because for 10 years ago I did not know that we can now sequence everything. We can sequence all bacteria, all pathogen, and the human completely for cheaper than whatever. But so we will see. But I think
So, well, actually, the last question was related with the. You mean the religion is about the halal and haram? As you know, in Indonesia, the majority is a Muslim, and we always consider about the content and the of what we take for medication as well as for the vaccination. But for the genome editing, as mentioned by Dennis, because even now the researchers that has been conducted the research, they are more into the microbial genome editing, and we still don't know whether in the next 5 years we haven't got into the human editing. I think there will be a lot of discussion about that because Hai Jianku from China. Have you heard that about the editing? The only one guy who tried to do the human editing, but I think still a complexity about that, and there are still a lot of controversial things that need to be discussed. So, yeah. So I don't know, and I don't think we can right now already decide whether it's related with the halal or the haram thing because it is not yet applicable even to human. So we are still. I think all of researchers are still doing the research about that. Yeah. And as maybe Dennis mentioned that we cannot guarantee maybe in the next 10 years whether that applicable because sequencing, as you said, now is really cheap even to sequence the whole genome for any species. Yeah, I hope that will satisfy your question. Yeah. Okay. So we will go to the next question. I think from the students, yeah, and from Prof. Erlia. I think she's doing a lot with the bacteriophage. Yeah. Anybody can help me to give yes. And then the next one to Prof. Erlia. Yeah.
Okay. First of all, thank you for the.
Yes, please. Just try to speak, and then he will turn on.
So actually, my question is kind of the same with the previous question. But, maybe I want to ask the direct impact of the base up and the direct impact that. What will the future plan that we have to prevent any outbreak in Indonesia? Maybe if we want to specify it more, it can be in Jember. Can I get an example of a direct plan towards preventing any outbreak, especially in Jember?
Especially in Jember, they are just arrived in Jember, but okay. Yeah, okay. Is that also, you asked about the direct plan to prevent the outbreak in certain region?
Yeah.
For example, like in Jember. Outbreak about what? A general outbreak of infectious disease, or do you have any specific request for specific disease?
Okay. maybe because previously we were talking about hepatitis, right?
Yeah,
And
hepatitis.
as we know, in early 2020, I think there's an outbreak of hepatitis A.
Yes.
Yeah. Maybe
Yes.
we can talk about the hepatitis.
Yes, the hepatitis is an annual problem in JEMBER. Yeah. Okay. Yeah. Okay. What is your name? Can you maybe?
Okay, I'm
Yes.
sorry. My name is Yordan.
Huh?
Yordan.
Yoda from medical faculty.
Or from the medical
Oh, okay.
faculty.
Yes. Okay. Thank you.
Okay. Thank
Yeah.
you.
Okay. Yeah. Prof. Elia, the next one. Langsung aja lah. Tolong Pak.
Thank you, Prof. Kartika. My question addressed for Prof. Dennis.
Can you please introduce yourself first?
My name is Erlia, and I'm from Biology Education Study Program. Mostly my research is about bacteriophage, Prof. And you mentioned a bit about food therapy. In your opinion, how big the potential synergies between bacteriophage therapy and conventional antibiotic in addressing AMR? Thank you.
I think it's clear. Yeah. Okay. Yeah. Please, Philemon, for the first question.
Yeah, to the question, hepatitis A outbreak or any given outbreaks. Outbreak is an outbreak. First, we need to identify what causes, or is this an outbreak related to what kind of the pathogen, and to understand where it's the source so that you can contain the transmission per se, that the outbreak does not become an epidemic beyond the outbreak region, moving more and more, and so on and so forth. So what we do, what are the measures that should be put in place? So this is one of the activities of PACE UP, for instance. So the Pan-Asian Coalition for Epidemic and Outbreak Preparedness during crisis is one of the key elements what PACE UP is aiming to achieve. So we need the capacities in various forms, which means like, as we have known that during COVID, we do not know, we even do not have masks. In Europe, we do not have masks, we do not have the swabs available, and we don't have the PPEs or the personal protective equipments in place. And physicians do not know what I should treat this patient with. What is the mode of treatment, how this patient should be treated, for instance. So you have to increase the capacities needed in terms of diagnostics, in terms of treatment, in terms of stopping the spread, and a lot in terms of different capacities or multidisciplinary capacities. This has to be raised. Therefore, these capacities which are raised or used during the period of outbreaks to react and to respond to an outbreak situation. If we find a kind of Ebola-like a viral hemorrhagic fever, okay, we just have a response team which is being sent with a mobile laboratory to this place to have this infection being characterized and confirmed to be an infection of biosafety level 2 or 3. Therefore, we know how quick or how contagious is the spread of this virus per se. And equally, what is important is you have to increase the awareness also from the community. Please avoid eating any wild animals, meat and things, bush meat and things. The awareness has to be equally increased in the society so that such a spread could be avoided, because individuals per se by themselves are also reservoirs or causes for such outbreaks. I can tell you one example in South Korea. One individual in the South Korean Sinjeon church, patient number 31, during the COVID-19, she was infected. She went to the church of 3,600 disciples, and one person one person made 64% of the whole CDC cases in South Korea. So this is a cluster of cases. It's called South Korean cluster. Such wise you have an Italian cluster. One guy spreading to the whole of the hospital, and the whole hospital was closed. So these clusters can be avoided if we could have an early detection. To have an early detection, we need the capacities to be put in place. Thank you.
Yes, 11, very well formulated. So early detection, so early actions to avoid. So not even outbreak, but to avoid is better. Yeah. Okay, and the next question for Dennis. Yeah, please, Dennis.
Yeah, sure. But maybe I can add to what Belavan said, because what we're doing now in PACEUP in Vietnam, for example, building the capacity to do surveillance. So actually the key point is surveillance. So if you do active surveillance, you can see, for example, what is your average, detection of a certain thing. For example, if it's 0 or if it's whatever, and then you can see if you do constantly surveillance, you can see the different peaks. And if you see that something is increasing, the first alarm would be that thing. So before it goes really big, you can already identify. So this is why I think a lot of effort is being put into building the capacities, building laboratory capacities, doing genomic, for example, genomic characterization, even for pathogens, for virus, bacteria, for everything. Because if you can see, some kind of deviation from the norm, then you would know, okay, we need to do something before it becomes really big. So I think that is the core of PACEUP, I think, also, right, in a lot of settings. So. And then going back to the phage, the phage is actually very interesting because in some of the some European countries actually already been used, like in Georgia, for example, in a lot of I think in Russia and in Belgium even. In Germany, we actually neglected it for a long time because of this ethical question, and also the challenge. For example, now it's starting, so there is a big dynamic going on. I think for the treatment of local infection, like if it's skin infection, I think it's fine to do that. If it's a systemic, this is becoming into the ethical question, what is it doing with the whole body? We don't know. Of course, it is actually specific for a certain pathogen, but the microbial community is quite large, so, and we cannot predict what is happening with the whole gut, for example. But the challenge actually with using this at the moment is the GMP level production, because before it can be used, it has to be used like that, and then also the testing, because the phages are very bacterial specific, so you cannot really take always the same phage to treat one species, so you always have to test a few and then make a phage cocktail and then treat them. And I think that is a challenge, but like you said, there is maybe an opportunity by combining antibiotic and phage together, because I think that is quite interesting. Then you would maybe use less antibiotic and then treating it faster, and that means the overall pressure, I think, will reduce. So I think that will be probably one of the ways of the future. So there are a few efforts, I know from a few groups who are working on urinary tract infection, so combining phage and antibiotic. But it's still at the moment in the experimental, so it's not in human yet. So it's just on the in vitro level. Yeah.
I hope it is going. Do you want to give a feedback, a question of everyone? Yeah, for the next, he wanted to have a further collaboration on that because he's working a lot with the phage and you're working a lot with the bacteria.
Yeah, so and actually in Germany there is this German network for infection research, and they are now very interested in this. So I think there is a lot of opportunities. I think it's quite interesting also. Yeah. Yeah. Sure.
Thank you very much. Yeah, thank you very much. The student that with Yoda, is that okay, or do you need to clarify things more? Okay. So I would like to invite now for the next session from our student in the other class, because I saw just 5 minutes ago. Is there any question from all of you over there? Hello, is there any lecture staying? I think Pak Sugianta or yeah. Okay, yeah. We see now one student. Yeah. Please mention your name and yeah. Go ahead.
Excuse me, can you hear me?
Yes, clearly, very good.
Okay, thank you. So my name is Edelweiss, and I'm a first-year medical student here. So for my question is, actually, it always fascinates me how, like, we as a human from the beginning of time, we always find ways to solve problems. But maybe I'm looking way too far into the future, but maybe, is it possible for us as a humans to run out of ways to cure like infectious diseases? Like, for example, as we discussed earlier, as time goes by, there are more pathogens, microorganisms that resistant to antibiotic. And so what are the most possible plan that we currently have to prevent this from happening? I think that's all for me. Thank you.
Thank you very much for the question. Yeah, I think I will just give over directly to both of you.
Well,
Yeah,
from
please.
my perspective, they are repurposing some of those, or they're combining different antibiotics combinations, because getting an antibiotic into commercial market takes at least a minimum of 30 to 35 years after different phases. So given that we have only hardly 1 or 2 new class of antibiotics left for treatment for any given infections, we have to use the permutations, combinations with the different antibiotics.
Yeah, it's difficult. So actually, what you said is actually the worst case scenario. And if we will ever come to that point, my answer is we are already at that point. So actually, we do have some patients already where we cannot do anything. So the thing is, they're mostly, they have an infection and everything is resistant. Really, every single antibiotic which we have is resistant. We call them a pan-resistant bacteria. And sometimes you cannot do anything actually. So the only things we can do supportive, we try to combine antibiotics, although we know that it's not going to work because you cannot do nothing, in the hope that by combining maybe it will work better. But for most of the cases, actually it would fail. So we do have a lot of these cases. But these patients are usually severely ill, and thankfully this does not come very often. But I think in this year we already have like 3 or 4 of them. So because at the moment, because of the, we have also patients from Ukraine, for example, due to war injuries and everything, and most of them are infected with Acinetobacter or something else, multi-drug resistant bacteria. In those patients, we actually do already see a lot of pan-resistant bacteria. So I think to answer your question, like we are already there actually, but thankfully it's not so common yet. I don't know if you see it here as well in Indonesia, but because sometimes if you do not look for it, you do not find it.
So, the message is to look forward to it.
Yeah, search and destroy.
Okay, thank you, Dennis. Yeah, I think we still have like 10 minutes for one more question. Yeah. Two more persons. Okay, that's fine. Yeah, anybody can help? Hand over the mic. Yes, di depan Pak. And then the next one will be the student behind, yeah? Please, could you please introduce yourself, yeah?
Thank you for the opportunity, and my name is Ina.
Test.
Thank you for the opportunity to ask a question. Maybe my name is Ina. Maybe it's a short one question. How is the chance that someone with hepatitis E infection will develop hepatocellular carcinoma? Thank you.
It's a very sharp question. Yeah. Are you going to answer directly?
Well, I could do
Yes,
that.
yes, okay.
So it depends on if the patient is immunosuppressed or if he is really competent, because normally most of these infections are self-limiting. You are asymptomatic self-limiting because you will clear this infection at some stage. If you have any other underlying comorbidities, or you are under immune suppression, or you are at high risk groups, then this may lead to a fulminant course of infection. In terms of percentage, at least 1 to 3% of the people goes into this fulminant course of infection.
Okay. Is that clear to Ina?
Well, so I know that hepatitis B can go into the hepatocellular carcinoma, but is it also the hepatitis E, as you say, that there is an additional condition that it
Sure.
needs to be.
So all hepatitis makes or leads to liver cirrhosis or hepatocellular carcinoma. It's one stage, like chronic hepatitis to liver cirrhosis to hepatocellular carcinoma or liver cancer per se. Hepatitis E also does this, all hepatitis including C, A, B and so. But the intensity or the clinical prognosis from an acute to hepatocellular carcinoma is much low compared to that of the hepatitis B, because the replication is much more faster and quicker and virulence is much higher in terms of hepatitis B and C compared to A or E, because A and E are RNA, B is a DNA virus.
Alright, so the chance is lower than hepatitis B. Thank you.
Okay, now the next question, please. Yeah. You hand over the mic, please. Yeah, thank you.
Thank you for the time, Prof. My name is Norsiki Mamadi. I am student in Pharmaceutical Science Magister in University of Jember, and I want to ask for Prof. Dennis and about strategy for reduce resistant antibiotic. Because in Indonesia, facility in most hospital, I mean, is not support for detection type of microbial or so what should we do if we are to combination of antibiotic? On my experience, Prof, I find some patient in hospital with some antibiotic but not yet full recovery. Mungkin itu. Terima kasih.
Thanks.
Thank you.
Okay.
So,
Yeah.
so I repeat it quickly. So it goes along the line on giving the right antibiotics, right? So something like that, or is it?
Yes.
So yeah, that is the thing. Actually, I can maybe share a personal experience which I also had discussing with a few friends and families and also doctors in Indonesia. Sometimes they do not really do diagnostics. This is actually the problem.
Yes.
For example, just for example, I know like a little kid, 5-year-old, has a pneumonia, and the doctor said, "Oh, it is viral. Don't do anything. Just stay at home." And then a few days later, this kid cannot walk anymore because he was really sick. And then the doctor said, "Of course, let's go to the hospital. Let's start with meropenem." And I said, "Wait a minute, that cannot be correct." And I said, "Did you already do diagnostic?" They said, "Oh no, we don't need it. Meropenem is broad spectrum, it will work." And I said, "Yeah, but this is a community-acquired pneumonia. And if you think about the etiology, what kind of bacteria or virus is causing this, it's not the gram-negative, the problem, which is probably pneumococcus or something. And by meropenem, of course, it will not be covered by meropenem because it doesn't work against gram-positive so well." That is one example, but at the end we did a PCR and this kid actually has Streptococcus pneumonia, and then of course meropenem did not work and it has to be changed. So I think a lot of just as an anecdote or as an example, I think a lot of the efforts cannot work if we do not have a strong diagnostics. I know that some countries do maybe over-diagnostic, so there is it's difficult to find the sweet spot because diagnostic is also expensive. But if you do not do diagnostic, then it's really difficult to do like a guided therapy, because I think that is one of the key actually into reducing antibiotic use, at least for the clinical medicine, to do diagnostic and then to do fast diagnostic. Because if you do diagnostic and if it takes like 5 days and the patient dies after 3 days, then it doesn't make any sense as well. So I think a lot of the capacity building would have to be also creating the laboratory capacity, but also creating the awareness in the doctors and also in I think in also the medical studies that that diagnostic is important. That microbiology diagnostic is actually not something that you do if nothing else works, but you do it before already, so to start. And I think I don't know if you know it, because when I was here as a kid, my doctor or my mom also told me like, "Oh, if you get an antibiotic, you have to take it as long as you can, whatever, like take it until this is finished." Nowadays, we're kind of also rethinking, do you really need to do that? Like, for a lot of infection, you can already stop after 5 days. So I think it's a lot of effort for you guys, actually the younger generation, to kind of drive this forward actually, because I think a lot of I would say the ideology and the dogma in the medicine is actually so strong, like from the older generations. I think it's up to you guys to kind of change it. So this is what we also try to do in Europe, and it's not so easy because, of course, it's been established for a long time, but it's working. So I think it's a good question. Yeah.
Okay, Velavan, do you want to give some comments on that?
Well, that's a good example what he has given. That's fine. I mean, you need to start somehow an empirical therapy. This is clear, but continue with the diagnostics in parallel, and equally use the diagnostic results to change the therapy regimen. It's not like anyone coming with a lower platelet count is a dengue, for instance. Yeah.
Can I also ask the question? Am I allowed to? Do you have antimicrobial stewardship program actually in the clinics here? Antibiotic stewardship program?
Hello, Ulva.
Do we have actually? Do you have an antibiotic stewardship program in the hospitals here actually? Antibiotic stewardship.
Stewardship, which is like how to use antibiotics.
I have a pick for the next Space Up workshop.
Okay.
Antibiotic stewardship.
Okay.
No.
Because I think that term is not so familiar
Yeah.
here. Can you please maybe take an
Okay.
Indonesian term on that?
The Indonesian term is I don't know. Yeah, I think it's something similar. So like it's like a group usually, a consultation wise. So you would have a look, and then what we do, for example, is we look at every patient, like severe patients, for example, and we review if everything is done. Like is it diagnostic done? Is anything else that can be done? Is the empirical therapy also before the results are there? Is it correct? Is it really covering every pathogen? And once also that finished, we follow up. Then we say, okay, this has to be changed into this, or something which is, at the beginning you always start to go broad because you also want to, get rid of the bacteria or any other pathogen. Sometimes once the microbiology is done, you can say, oh, this actually maybe amoxicillin susceptible. Then you give amoxicillin instead of ceftriaxone, which is more broad. So that's reducing the burden. So we try to what we call de-escalate the therapy, so to step down, and to also review can we maybe give this shorter? Do we really need to give it for 14 days? Can we just do it for 5 days? So I don't know, maybe there's a different terminology in Indonesia, I don't know, but what we use is the so-called antibiotic stewardship or antimicrobial stewardship.
Stewardship.
But when you go to the clinics and also with the you see the patient, and then this is very similar actually. So I think that's one of the important things to do. However you sell it as a program, we sell it as a stewardship program, but I think it's important, and I think it's quite interesting because I think that way you can also make a difference, even if you're working in the lab. So this is what I think, because I worked in clinical medicine in the beginning, but then I now move into more lab. But I'm still very interested in patient care, so I think it's always good to be able to contribute something, because you have knowledge which you can share actually.
Denise, I'm sorry, I have to interrupt you. I know that the discussion is getting warmer and getting hotter, but we still have some time until tomorrow evening. If you wanted to ask more questions on specific topic to Velavan and Denise, they are still here. But for this session, I'm very sorry that I have to close this session because we are kind of 5 to 10 minutes too late from our schedule. But before we really end this session, I would like to kindly ask Velavan to give a closing statement, not in German but in English, please. And Denise, so you said you cannot speak Indonesian, but you are fluent in Indonesia, so I ask you to give a closing statement in Bahasa. A very very challenging thing. Yeah, please, Belavan.
Thank you. I mean, it was suddenly she was asking me what should I say as a closing statement. I mean, we are here to transfer our knowledge, experience, wisdom, and I mean, a lot of students, I mean, I see, you know, like 25 years or 20 years like you before. I'm trying to fit in your shoes. You know, the level of education and the level of knowledge we have at that age and the level of education and knowledge we have at this age is completely different. But things have changed in the last 20 years. I mean, we have been exposed to all this internet connectivity, a lot of reading. You are even better off in many things. But my thinking is like you are the next generation leaders or next generation budding scientists, and you are much better. You know, like I try to ask you, if you have a question in your mind, try to ask them immediately. If not, it remains as a question for your life. And equally, try to read a lot in terms of internet, but internet is also a kind of a curse and a boon, because your eyes will pick only what your eyes or your brain want to see. So the keywords, even if use of keywords is equally important. Read a lot, but then don't stop reading at one time point. Read it from the start to have a full picture, you know, not to stop it in between. So we are here, we will be here for the next, in the next 6 months' time with a different range of topics, not only to educate, but also we would like to spread the need and importance of such studies in terms of infectious diseases. So that's our goal. I mean, I have nothing much more to add here, but I'm really thankful for the whole team who has invited us here and giving us a very good hospitality. And we are indebted to this, and we look forward to contribute more, and the needs should be fulfilled. And please ask your teachers and professor colleagues to identify what are the priorities that you would like to learn. Thank you.
Thank you very much, Belavan. Now, so Dennis.
Coba ya, aku nggak tahu bisa atau enggak.
Jangan cepat-cepat ya, ngomongnya jangan cepat-cepat.
Nah, susah. Kalau terlalu lambat agak susah. Kebiasaan ngomong cepat.
Okay, please.
Ya mungkin kalau misalnya untuk closing statement, belajar bersama kayaknya mungkin ya harus apa? Kalau jadi kita harus sadar bahwa belajar itu adalah proses yang berjalan, dan nggak stop dalam satu sisi. Dan meskipun mungkin kita di Eropa punya cara yang lain untuk mengatasi masalah, tapi bukan berarti masalah ini cara kita itu cara yang paling benar. Itu mungkin sebetulnya kita harus mungkin something more like self-critical, harus bertanya apakah itu cara terbaik, dan berdialog dengan orang lain. Dan itu kayaknya adalah yang saya dapat dari simposium yang lalu, dan juga dengan melihat semua keadaan dan juga proses-proses di negara-negara yang lain itu sebetulnya menarik sekali. Jadi buat kita juga, kita belajar juga untuk ke depannya. Jadi ya mungkin closing statement belajar bersama dan kolaborasi.
So, Belavan, you ask the translation later on to Dennis, okay?
I will translate it in German for you.
Yeah, a very nice and very interesting session before. You know, some of our students have made some recording, I think, to your speech. Oh my goodness, so, you know, Indonesia is always like, always upload it to the social media. You just play later on. So, once again, thank you very much for Belavan and Prof. Dennis for today's session and lecture and discussion. And I kindly invite you all to give a very big and warm applause to both of speakers. Yeah, okay. From me as a moderator, thank you very much for your attention. And for the next session, I will just hand over to the MC, Bu Enda. Thank you.
Thank you, Bu Kartika.
to Professor Velavan, Professor Dennis, and Professor Kartika Senjarini. Dear ladies and gentlemen, now we have come to the end of this program. The committee would like to express our sincere gratitude and appreciation to all the speakers, the moderator, as well as all the participants joining fully from this morning. We do hope that all of you will have fruitful and productive discussion. Now you can see the QR code. Please, QR code for the certificate. Yes, now you can see the QR code for the certificate of attendance on the screen. Please select which certificate that suits you with SKP ID and IAKMI or without SKP. Finally, the committee would like to deliver apology for any inconvenience. We hope to meet you again in the future. Thank you very much for your attention. Have a nice day. Wassalamualaikum warahmatullahi wabarakatuh. Once again, Karawitan Krito Laras Budoyo from University of Jember.